Hello students and welcome to the study portal. The topic of today's video is analytical technique. So in today's video we will understand about what is analytical technique and how analytical techniques are used in the laboratories to determine the chemical or physical property of a chemical substance or a mixture. So without delay, let's start the video. So analytical technique is a method which is used in the laboratory to determine the physical or chemical property of a chemical substance, chemical element or the mixture. Alright, so there are variety of techniques which are used for analyzing the chemical or physical property of elements. They can vary from simple weighing to advanced techniques which are performed by using the highly specialized equipments or instruments which are present in the laboratory. So it is a method to determine the concentration of a chemical compound, we can say. The analytical techniques are the methods which are used for the qualitative and quantitative determination of concentration of a compound by using various techniques like titration, spectroscopy, chromatography, electrophoresis and gravimetric analysis. So they use the concept of qualitative and quantitative determination of the concentration of any chemical compound or substances. So basic and most widely used analytical method techniques include the electrophoresis, chromatography, spectrophotometry, ELISA and the different techniques. So in today's video we will gonna cover about the electrophoresis, the chromatography, spectroscopy, ELISA and different techniques that are used in the laboratory for the diagnosis of these chemical compounds. In-depth knowledge of analytical laboratory technologies and how to apply them to a specific sample is critical to driving understanding about a new chemical formulation or product during development across many sectors. Precision analytical technologies are required to assess production quality and to determine trace level impurities which may present a risk to human health or the environment. These technologies are often highly specialized analytical instruments which can only be operated by the technicians who have industry application experiences. So here are some of the analytical techniques that are widely used in the diagnostic laboratory. So let's have a look. First one is the chromatography, second one is the electrophoresis, third one is the spectrophotometer, fourth one is the centrifugation and the last one is the enzyme linked immunosorbent assay which is called as ELISA. Let's talk about chromatography. As the name implies, chrome means color and graphy means a graph or a representative graph of the color of the substance. So chromatography is an analytical technique which deals with the separation of closely related compound from a mixture, such as we have to separate protein, peptides, amino acids, lipids or carbohydrates present in the serum sample. So for that technique we will use the chromatography technique. So chromatography is usually consists of a mobile phase and a stationary phase. Mobile phase means mobile means moving. So mobile phase refers to the mixture of substance that has to be separated, which is dissolved in liquid or gas, and the stationary phase means which is stationary, which is not moving. So it is a porous solid matrix through which the sample contained in the mobile phase percolates. Alright. Now the interaction between the mobile and stationary phase results in the separation of compounds from the mixture. In chemical analysis, chromatography is a laboratory technique for the separation of mixture into its components. The mixture is dissolved in a fluid solvent called the mobile phase, which carries it through a system on which a material called the stationary phase is fixed. Because the different constituents of the mixture tend to have different affinities for the stationary phase and are retained for different lengths of time depending on their interactions with the surface sides. The constituents travel at different apparent velocities in the mobile fluid, causing them to separate from the mobile phase. The separation in the chromatography is based on the differential partitioning between the mobile and the stationary phases. The subtle differences in a compound's partition coefficient results in differential retention on the stationary phase and thus affects the separation. So, the chromatography may be preparative or analytical. The purpose of preparative chromatography is to separate the components of a mixture for later use and is thus a form of purification, we can say. But on the other hand, analytical chromatography is done normally with smaller amounts of material and is used for establishing the presence or measuring the relative proportions of analytes in a mixture. There are different type of chromatography that is used in the diagnostic laboratory. First one is the paper chromatography, second one is the ion exchange chromatography, and third one is the affinity chromatography. There are different types of chromatography are also used in the laboratories such as TLC, which is thin layer chromatography, and GLC, which is gas liquid chromatography. Now talking about the paper chromatography. So, paper chromatography is useful for separating the mixture of amino acids, sugar, chemicals, lipids, urea and some other drugs. As the name implies, the paper chromatography is performed on the filter paper, which is used to support a stationary water phase, while the mobile organic phase moves down the suspended paper strip in a cylinder. So, the separation in the paper chromatography is based on liquid-liquid partition of the compound. Thus, it is very important form of partition chromatography between two liquid phases, in which first one liquid phase get absorbed on the surface of the paper and the other moves through the filter paper, which is suspended in the cylinder. Paper chromatography is generally used to separate low molecular mass compounds based on their distribution between the stationary phase and the liquid phase and the mobile phase. Due to its low cost and availability of various protocols for the separation of compounds, paper chromatography is considered a powerful analytical tool. Alright, so it consists of two-dimensional chromatography, which involves two solvents and rotating the paper 90 degree in between. This is useful for separating complex mixture of compounds having the similar polarity. For example, the amino acids in the serum sample. Now, let's have a look at the procedure of the paper chromatography. So, what we do in paper chromatography, let's understand. So, in paper chromatography, a horizontal line is drawn near one end of the paper. The sample needs to be separated is placed as a small drop or line onto the paper using the capillary tube. Labeling the drop by a pencil with an alphabet or number so as to help to identify the component later. Thereafter, the paper is then placed into a sealed container with a shallow layer of suitable solvent. The solvent level must be lower than the pencil line or drop on it. The container needs to be covered to stop the solvent to evaporate. Thereafter, the solvent rises up the paper chromatography, taking each component of the sample with it. The components travel with the solvent depending on three things. First one is the polarity of the sample molecule. The non-polar molecules or the components travel faster than the polar components. Second one is the attraction between the sample molecule and the solvent or solvent mixture. And third being the attraction between the sample and the silica. Let's understand with the help of an example. Suppose any sample compound mixture contains three colored molecules, green, blue and red. According to their polarity, the order of these components is red is the least, then blue, then green. Thus, the most non-polar green will travel first along with the mobile phase, then blue, and at last most polar compound, the red one. Alright. So, when the solvent rises near the end of the paper should be taken out from the sealed container and should be air dried. The paper with separated bands of components are then observed under UV light. So, this is the whole procedure of the chromatography. So, the rate of migration of molecule depends upon the relative solubility in the stationary phase and the mobile phase. Now, how we identify the compound? The distance travelled by particular component of a mixture is used to identify it. We identify generally by resolution factor. So, what is resolution factor? Resolution factor is the ratio of the distance that the compound travelled and the distance that the solvent travelled. Alright, in mathematical expression, you can see here it is written that the resolution factor is equal to the distance from origin run by the solute and distance from origin run by the solvent. Here you can see it. So, paper chromatography is a chromatographic technique which is used to separate mixture of chemical substances into its individual components. Alright, now talking about the significance of the paper chromatography. So, it can be used for separation of components of the ink because it is based on the colored movement of colored substances on the basis of their polarity. Next one is the for separation of amino acids which is present in the serum or any kind of sample. Then to detect any kind of adulteration in foods and drink, we can use paper chromatography, and to determine the component or the value or the concentration of drugs in humans and animals, we can also use paper chromatography. Now talking about ion exchange chromatography. It is also widely used in the laboratory. It is used for the separation of amino acids, protein, nucleotides and the charged molecules. So, as the name implies ion exchange, it is based on the exchange of the ions between the molecules on the basis of their electric charges. So, the ion exchange chromatography is based on the principle of separation of molecules on the basis of their electric charges or the electric charges they possess. So, in ion exchange, what happens? The resin cation exchanges exchanges with the anion exchange, and thus the separation of the chemical components takes place. The ion exchange chromatography works on almost any kind of charged molecules including large proteins, small nucleotides and amino acids, but it must be done in conditions that are 1 unit away from the isoelectric point of a protein. Alright, so there are two types of ion chromatography, for anion exchange and the cation exchange. Cation exchange is used when the molecule of interest is positively charged, and anion exchange is used when the molecule of interest is negatively charged. Now talking about the procedure. So before ion exchange chromatography can be initiated, first it must be equilibrated. The stationary phase must be equilibrated to certain requirements which depends on the experiment that you are working with. Once the equilibrated, the charged ions in the stationary phase will be attached to its opposite charged exchangeable ions. The exchangeable ions such as chlorine or sodium, etc. Next, a buffer should be chosen in which the desired protein can bind to. After equilibration, the column needs to be washed. The washing phase will help remove all impurities that does not bind to the matrix while the protein of interest remains bounded. Thus, in ion exchange chromatography, ions in solutions are reversibly replaced by ion exchange resin, and the binding abilities of the ions bearing positive or negatively charged is highly dependent on its pH, because the net charge varies with the pH. Here in this picture, you can see a negatively charged analyte and a positively charged analyte. So, a positively charged analyte is attracted to the negative surface, which is cation exchanger. We can call it as stationary phase particle, and a negatively charged analyte gets attracted to the positively surface. Thus, the exchange of ions is carried out, which results in the separation of ions and polar molecules based on their affinity to the ion exchanger. Now talking about the significance of the ion exchange chromatography, it is performed for the separation and purification of many charged or ionizable molecules such as proteins, peptides, enzymes, nucleotides, DNA, antibiotics, vitamins, etc. Now understanding the affinity chromatography. So affinity chromatography is used for separating biochemical mixtures. The principle is based on the interaction between antigen and antibody or enzyme and substrate or receptor on gland. Basically, it is based on the property of specific and non-covalent bonding of proteins to other molecules, which can also be called as ligands. For example, we know that enzyme bind specifically to ligands, which can be a substrate or the cofactor. So, the affinity chromatography is based on the property of the substance to which it is binding with. So, the principle of affinity chromatography is that the stationary phase consists of a support medium, we can say cellulose beads or other, on which the substrate, sometimes it can be a coenzyme, has been bound covalently in such a way that the reactive groups that are essential for enzyme binding are exposed. As the mixture of protein is passed through the chromatographic column, those proteins that have a binding site for the immobilized substrate will bind to the stationary phase, while all other protein will be removed in the void volume of the column. So, this is the principle of affinity chromatography. The procedure of affinity chromatography involves the use of ligands which are covalently attached to the inert and porous in the column. So, the two phases of an affinity chromatography are the same as paper and ion exchange, the mobile and the stationary phase. First, we have to add silyl lysate to the column. The protein of interest will interact with the cellulose beads through the matching affinity. Then we will add wash buffer and remove remaining unspecific protein and other substance. Then we will remove or separate a protein of interest from the affinity beads through an elution buffer. Either via pH exchange or salt concentration, the protein of interest will separate out. Then we will purify an active protein of interest. Alternatively, some reagents that can break protein ligand interaction can also be employed for the separation of proteins. So, this is how it is performed. A ligand is present in the cell column. We mix analyte with this. The analyte pass from an affinity column. Then the analyte is retained in the stationary phase, and then we will elute analyte from the sample with the help of cell beads. The significance or importance of affinity chromatography is that it can be used for isolation and purification of all biological macromolecules. It can be used to purify nucleic acid, antibodies, enzymes, etc. It is used to notice or separate biological compounds that bind to a particular substance, that is a particular enzyme can be separated, and to reduce amount of substance in a mixture. Next is HPLC, which is also called as high performance liquid chromatography. It is used to separate components of mixture by using variety of chemical interaction between the substances. The separation principle of HPLC is based on the distribution of the analyte between a mobile phase and a stationary phase. Depending upon the chemical structure of the analyte, the molecules are retarded while passing the stationary phase. The specific intermolecular interaction between the molecule of a sample and the packing material define their time on column, and thus different constituents of a sample are eluted at different times. Thereby, the separation of the sample ingredient is achieved. There, a detection unit, for example UV detector, recognizes the analytes after leaving the column. The signals are converted and recorded by a data management system or computer software, which is then shown in a chromatogram. After passing the detector unit, the mobile phase can be subjected to additional detector units, a fraction collection unit, or to the waste. In general, HPLC system contains a solvent reservoir, a pump at high pressure, a constant speed through system to keep the drift and noise of the detector signal as low as possible. A constant and pulseless flow from the pump is crucial. Then a detector to detect the substance, and the PC for data acquisition, and waste container for removing out the mobile phase waste. The significance of HPLC is that it can be used for chemistry and biochemistry research for analyzing complex mixture. It is used for purifying chemical compounds. It is used for developing process for synthesizing chemical compounds and to isolate natural products or predict the physical properties of a natural product. It is used to quantify assays of final products, to evaluate the product stability and monitor degradation. So that is all about today's session. In today's session, we concluded about the chromatographic techniques, chromatographic analytical techniques that are commonly used in the diagnostic laboratories. We will come with next part of this video, in which we will cover about the electrography techniques or other analytical techniques that are used in the laboratory, such as spectrophotometry, ELISA, and many other. Thank you, students. Hello students, and welcome to the study portal. The topic of today's video is Analytical Technique Part 2. This video is in continuation with the previous video, in which we covered about introduction to the analytical techniques, and few techniques that are used in the diagnostic laboratory for analytical techniques such as chromatography. We discussed about the different types of chromatography such as paper chromatography, ion exchange chromatography, affinity chromatography and the high performance liquid chromatography. In today's video, we will discuss about few other analytical techniques that are used in the diagnostic laboratory for the chemical analysis of a compound. So, let's start the video. So what is electrophoresis? We will discuss about the electrophoresis in today's video. So electrophoresis comes from a Greek word which means to bear electrons. Alright, it means to carry the electrons. It is a laboratory technique which is used to separate different elements of cell or any compounds such as DNA, RNA or protein molecules based on their size and electrical charge. The movement of charged particles in an electric field results in the migration or movement of the ions to the oppositely charged electrode. This process in which the ions migrate and thus they are separated from other elements is known as electrophoresis. In electrophoresis, an electric current is used to move molecules to be separated through a medium. Alright, so there are different types of electrophoresis: paper electrophoresis, gel electrophoresis, isoelectric focusing, page. So these different type of electrophoresis techniques are used in the diagnostic laboratory for the separation of compounds. In addition, I would like to tell you that the electrophoresis of positively charged particles or cations is sometimes called cataphoresis, while electrophoresis of negatively charged particles or we can say anions is sometimes called anaphoresis. The electrokinetic phenomenon of electrophoresis was first time observed by Russian professor Peter Ivanovich Strakhov and Ferdinand Frederick Reuss at Moscow University in 1807. They noticed that the application of a constant electric field caused clay particles dispersed in water to migrate, which lends them an idea that the movement of ions is occurring in the presence of the electric field. So, it can be used as a basis for analytical techniques used in chemistry for separating molecules by size, charge or binding affinity. Electrophoresis is used in laboratories to separate macromolecules based on size. The technique applies a negative charge, so proteins move towards a positive charge. So basically, the charged particles in the presence of electric field move towards the oppositely charged electrodes, which results in the separation of these macromolecules. Now talking about the different types of electrophoresis. So the first type we will discuss is the paper electrophoresis. As the name implies, paper electrophoresis is carried out on a filter paper. Alright. Now the technique of paper electrophoresis is simple and inexpensive, and it requires only micro quantities of plasma for separation. The support medium that we use in paper electrophoresis is a filter paper. This filter paper is wetted with dried buffer solution. The electrophoresis apparatus in its simplest form consists of two troughs to contain buffer solution through which the electric current is passed. The paper electrophoresis is frequently used for isolation of proteins, amino acids and oligopeptides in the plasma. So in paper electrophoresis we can say the separation of charged particles is carried out based on the differences in their migration rate. The migration rate can be varied because of its electrical charge, size of particles and the shape of particles. Let's talk about the procedure of the paper electrophoresis. In paper electrophoresis, the sample, whatever sample we are taking, such as we are taking plasma sample for the separation of proteins. So the sample is applied on a strip of filter paper wetted with dry buffer solution. The filter paper is wetted into dried buffer solution. Now what we do, at the end of the paper strip, we dip it into the buffer reservoir in which electrodes are placed. Then electric current is supplied, allowing the molecules to migrate for sufficient time. On the basis of this migration of the compounds, the different compounds or different molecules are separated. Then the paper is removed. As you can see in this picture that a filter paper strip is there. It is wetted into dried buffer already. Then we will dip into we will put the sample, the plasma sample for example we are taking, and we will dip it, the ends of the paper, into two electrodes. Alright. These are the buffer reservoirs in which the electrodes are put. As you can see, 2 electrodes are put into it and the electric current is passed. So now what will happen? The compounds present or the molecules present in the sample will move in the presence of electric field as per the size or their affinity or the shape of particle. For the separation of serum proteins, Whatman number 1 filter paper is used and press buffer is used, which has a pH of 8.6. The stains amino acid amido black or bromophenol blue are used for this purpose. Now what is the significance of electrophoresis? Electrophoresis is generally used for the separation of macromolecules based on their size. This technique applies a negative charge, so protein moves towards a positive charge. It is used extensively to separate or for the analysis of DNA, RNA and protein. Now talking about gel electrophoresis. As the name implies, gel electrophoresis is used with a gel. It is generally used for separation of proteins and nucleic acid. In this technique, the separation of molecules is carried out on the basis of their charges in addition to the electric charges. It is a laboratory method. In this technique, the molecules to be separated are pushed by an electric field through a gel which contains small pores. The molecules travel through the pores in the gel at a speed that is inversely related to their lengths. This means that a small DNA molecule will travel a greater distance through the gel than will a large DNA molecule. So in this technique, as you can see in this picture, that the process is carried out in a gel. Generally agarose gel is used as a stabilizing media. This gel contains wells which can be made with the help of a comb. Buffer is added into the apparatus. The sample from which the molecule has to be separated is loaded, and then the electric current is switched on. The separated components can be identified either by labeling with a radioisotope or by UV visible spectrophotometer. Gel electrophoresis involves an electric field. Let's understand this deeply. In particular, this field is applied such that one end of the gel has a positive charge and the other end of the gel has a negative charge. Because DNA and RNA are negatively charged molecules, they will be pulled towards the positively charged end of the gel. Proteins, however, are not negatively charged. Thus, they will move towards they are not negatively charged, they are positively charged, so they will move towards a negatively charged electrode. This means that the proteins with a detergent called sodium dodecyl sulfate will be mixed. So after the separation of components, they can be how will they be identified? They can be identified when you will label them with a radioisotope so that their color will be different, or when you will see or observe these molecules under UV visible spectrophotometer only. Now talking about the clinical significance or the importance of gel electrophoresis. So gel electrophoresis is used for molecular weight for estimation of proteins and nucleic acid, and for the determination of subunit structure of protein, amino acids, the purification of isolated proteins, or monitoring changes of protein content in body fluids. It is also used to quantify proteins, and it has some applications in the plotting technique also. Now talking about isoelectric focusing. Isoelectric focusing is used for the purification of proteins. The iso means same, electric means the charges. So in this technique, the all molecules are immobilized at the isoelectric pH. Isoelectric focusing is also known as electrofocusing. It is a technique which is used for separating different molecules by difference in their isoelectric point. Now what is isoelectric point? Isoelectric point is the pH at which a molecule carries no net electrical charge or is electrically neutral in the statistical mean. So at the isoelectric point where the pH of the molecule carries no net electrical charge, this process is carried out. It is a type of zone electrophoresis which is usually performed on proteins in a gel that takes advantage of the fact that overall charge on the molecule of interest is a function of the pH of its surrounding. So in this technique, stable pH gradients are used. They are generally set up in the gel which converts the pH range to include the isoelectric points of components in a mixture. As the electrophoresis occurs, the molecules migrate to positions corresponding to their electrical charges. As you can see that the molecules that have they will migrate or they will move towards their isoelectric point only, because the gel is made up of stable pH gradients of isoelectrical points. They get immobilized and form shape stationary bonds. As you can see in this picture, that sharp stationary bonds are formed. The gel blocks then can be stained and identified. The isoelectric focusing technique is used for separating proteins and polypeptides. It is also used to measure the density of band. It is used for research in taxonomy, cytology and immunology. It offers an effective alternative to conventional electrophoresis for genetic marker typing. So this is all about the electrophoresis. Electrophoresis is a technique in which the compounds are put into the under the influence of electric field, and under the influence of electric field, on the basis of their charge, size or shape, the separation of molecules is carried out. Next process under analytical technique is spectrophotometry. So spectrophotometry is carried out by this instrument called spectrophotometer, as you can see. It is an instrument which measures how much a chemical absorbs light. As we can see that spectrophotometer, photo means light, meter means measure, and spectrum means spectrum. So it measures the spectrum of light. So the intensity of light is measured as the beam of light passes through the sample solution. So whatever light is absorbed through that solution is measured by this instrument. So spectrophotometry is most commonly applied to ultraviolet, visible and infrared radiations. Modern spectrophotometers can also interrogate wide range of electromagnetic spectrum including X-ray, ultraviolet, visible, infrared and even microwave wavelength. But in general, the spectrophotometer measures the compounds or the molecules in ultraviolet, visible and infrared radiation. As we know that every chemical compound absorbs or transmits or reflects light over a certain range of wavelength. So spectrophotometry is a technique in which it measures how much a chemical substance absorbs or transmits. It is widely used for quantitative analysis in various areas of the diagnostic laboratory. Any application that deals with chemical substances or materials can use this technique. For example, in biochemistry, it is used to determine enzyme catalyzed reaction. In clinical, it is used to examine blood or tissues for clinical diagnosis. So there are different uses of the spectrophotometry. So spectrophotometry is carried out by an instrument called spectrophotometer, as you can see in the picture. It measures the amount of photons or the intensity of light which is absorbed after it passes through sample solution. With the spectrophotometer, the amount of a known chemical substance can also be determined by measuring the intensity of the light detected. Depending on the range of wavelength of light source, the spectrophotometer can be classified into two types. First one is the UV visible spectrophotometer, which uses light over the ultraviolet range of 185 to 400 nanometer and visible range 400 to 700 nanometer. And IR spectrophotometer, the infrared spectrophotometer, which uses light over the range of 700 to 15,000 nanometer of infrared range. So in a spectrophotometer there is a light source, there is a collimator or lens, then there is a monochromata, then wavelength selector is there, which is also called as slit. Then the sample solution is put into the cuvettes are there, and a detector is there which detects the amount of or intensity of light which is observed, and a digital display is there in which the concentration of the thing is, or the intensity or optical density of the solution is displayed. Now talking about the principle of spectrophotometer. So the principle of spectrophotometer is based on the Beer-Lambert's law. The Lambert's law states that the amount of light absorbed is directly proportional to the length or thickness of the solution under analysis. So it states that whatever amount of light is absorbed, it is directly proportional to the length or thickness of the solution. So I by 10 equal to E minus KB. Now Beer's law, what is Beer's law? Beer's law states that the amount of light absorbed is directly proportional to the concentration of the solute in the solution. So we can say that Beer-Lambert's law is a linear relationship between the absorbance and the concentration of a solution. So the Beer's law can be called as the concentration of the solution is directly proportional to the concentrate of solute or the light present in that solution. So we can say that the intensity of light that is absorbed depends upon the color of the solution or the light intensity absorbed by the solution. So more be the intensity of light absorbed, the more be the concentration of the solute present in that solution. Alright, hope this is clear now. Now talking about the significance of the spectrophotometer. So spectrophotometer is spectrophotometry is carried for the measurement of the concentration of any kind of solute present in the solution. It is used to detect the impurities present in any kind of solution. It is also used in chemical kinetics. It is used to detect functional groups, and molecular weight determination is also carried out from spectrophotometry. Now talking about ELISA. ELISA, also called as enzyme-linked immunosorbent assay, is a commonly used analytical biochemistry assay. It was first described by Engvall and Perlman in 1971. This is used to detect both current and the past or history infection. So it is based on antigen and antibody reaction. So it detects the amount of antigen present in the sample or the amount of antibodies that are present in the solution. This assay uses a solid phase type of enzyme immunoassay to detect the presence of a ligand in a liquid sample using antibodies directed against the protein to be measured. In the most simple form of ELISA, antigen from the sample to be tested are attached to the surface. Then a matching antibodies is applied over the surface so it can bind with the antigen. This antibody is linked to an enzyme, and then any unbound antibodies are removed. In the final step, a substance containing the enzyme substrate is added. If there is binding, the subsequent reaction produces a detectable signal. Most commonly, a color change is observed. So this is how ELISA is performed. So we can say that the ELISA works on the principle that specific antibodies bind with the target antigen and detect the presence and quantity of antigens binding. In order to increase the sensitivity and precision of the assay, the plate is coated with antibodies with high affinity. So, due to the presence of high affinity antibodies, the antigen binds, and in the last step enzyme substrate is given. If there is no binding, then if the antibodies doesn't bind, then it will be given as color change. So ELISA can provide a useful measurement of antigen antibody concentration. So ELISA is used for detecting antigens or antibodies or the concentration of antigen and antibody. ELISA was first test which is used for the detection of HIV. Now talking about the clinical significance. So it can be used for cancer screening to drug. It is also used for detection of HCG hormone present in the pregnancy. It is used for identification of cancer biomarkers for the early detection of cancer. It is used for the detection of platelet antibodies. It is used for food allergens. It is used for detecting viruses, and it is also used for Lyme disease, syphilis, etc. So the advantages of ELISA is that ELISA gives an accurate diagnosis of a particular disease. It can be carried out for complex samples as the antigen is not required to get purified to detect. And ELISA is highly responsible since both direct and indirect methods can be used. It is a rapid test which yields or which gives result very quickly and easily, and it is easier to perform, and it is not a complex process as compared to other types of techniques which uses the radioactive material. So it can be easily performed in the diagnostic laboratory. So the different types of ELISA are, there are three different type of ELISA. First one is the indirect ELISA, in which antigen is coated to the microtiter well. Second one is the sandwich ELISA, in which the antibody is coated to the on the microtiter well. And the third one is the competitive ELISA, in which the microtiter well, which is antigen coated, is filled with the antigen antibody mixture, and then there is a competition between the different antibodies to bind with the antigen. So that is all about ELISA. So ELISA has its application into different outbreaks also. As you can see in the COVID, to evaluate the spread of the disease, ELISA was carried out in the recent COVID-19 outbreak. Rapid testing kits are also used to determine the presence of antibodies in the blood sample. Now, concluding the matter of analytical techniques, we can say that analytical techniques provide an opportunity for researchers and scientists to explore advanced and latest research development. It can be used for separation of and identification of both macromolecules and the micromolecules. So we can say it is a wide range of techniques are used in the analytical techniques for the detection and the separation of biomolecules. So the constituent molecules from which macromolecules are assembled are called monomers, dimers or tetramers. So these subunit of the protein can also be diagnosed with the analytical techniques. So we can say that analytical technique is a method which is used to determine a chemical or physical property of a chemical substance, chemical element or the mixture. There are different techniques we studied about, such as chromatography, electrophoresis, spectrophotometry, ELISA and some other technique also. So the titrimetry also comes under the analytical technique method. It is the technique which is used to determine the concentration of the analyte. The analytical laboratory techniques and testing also provides the data which is required to make critical decisions during the research and development process, and it also improves the regulatory compliance in the life science also. So analytical laboratory technique is a very important part of the diagnostic laboratory. So you should you should have a deep understanding about the in-depth knowledge of the analytical laboratory technologies, that which analytical technique is used for the separation or the identification of different type of macro or micromolecules in the laboratory. So students, that's all for today. Hope you enjoyed the session. Keep learning, keep studying. Thank you.
In this video, we discuss about the analytical techniques that are performed in a biochemistry. So, what are the introduction of the analytical techniques? Actually, what are the analytical techniques? The techniques that are used in the biochemistry for the for the analysis of the different type of sample, for the different type of sample. That technique is known as the analytical technique. That technique is known as the analytical technique. So analytical techniques are the method used to perform the qualitative and quantitative determination of concentration of a compound by using various techniques like titration, spectroscopy, chromatography, gravimetric analysis, right? So these are the techniques that are used for the analysis of the sample or biochemical reactions in biochemistry. There are more different type of analytical techniques, like microscope is also analytical techniques, chromatography, not chromatography, actually chromatography is a separation technique, not for the analysis. Spectroscopy is a analytical techniques. So there are different types of immunochemistry. This all are analytical techniques that are all used in a biochemistry for the analysis of the sample. So classification of the analytical techniques. Analytical techniques are classified according to the qualitative and quantitative basis. In qualitative, first of all, what do you mean by qualitative and quantitative? Quantitative means when we measure something. What? When we measure a substance in a unit, measure in unit, that is known as quantitative. Qualitative means we measure according to their functioning, according to their properties, that is qualitative. So in qualitative includes XRPD, electrophoresis, TLC, IR, infrared. These four techniques are in qualitative. And in quantitative, ultraviolet spectroscopy, fluorescence spectrometry, HPTLC, HPLC, LCMS. Okay. Spectroscopy comes under quantitative, electrophoresis comes under qualitative. So remember this. So first techniques that we study in this video is the photometry. Photometry is the science of the measurement of light in terms of its perceived brightness to the human eye. Photometry, from name it is clear. Photo means, photo means, some problems. Photo means light, right? Metry means analyze the. So for the analysis of the or measurement, measurement. So for the measurement of light, that's come under photometry. Measurement of lights come under photometry. It is distinct from the radiometry, which is the science of measurement of radiant energy in terms of absolute power. Right? So photometry is the science of the measurement of light in terms of its perceiving brightness to the human eye. And next, it is the distinct from the radiometry, which is the science of measurements of radiant energy in terms of absolute power. This is the diagram. This is the diagram of the photometry. This is the light source. What? This is the light source. Clear? And this is the collimator lens. And this is the prism. What? This is the prism. And you know that when light go from the prism, the light was split in a 7 color. That is with geo. Right? The light is split into a 7 color. And from that 7 color, if you want to analyze a single color of the light, then there is a detector. That is a photocell. There is a detector, that is a photocell. We detect the light performed or intensity of the light source. Which color of the light, intensity of which color of the light is more, that are determined by the photocell. And according to the intensity, the value will be given or displayed. The value will be displayed on the digital display meter. Clear? The value will be displayed on a digital display meter. According to that value, we determine which type of light will be generated from the source. So what happened here? First, light source. From light source, light will pass from the lens. After that, the light will be split into a 7 color with the help of prism. According when 7 color will be splitted, when they split it in 7 color, the yellow color, or according to that, which color of the light will be present from the source, according to that only photocell decide the intensity of the light, and according to that intensity, the value will be displayed on the digital meter, and we get the color intensity of the light. So next is immune chemistry. What? Immune chemistry. From name it is clear. Immune means antigen and antibody. Immunity. Right? Reaction of antigen and antibody. So in this technique, we analyze the chemistry of the antigen and antibody reaction. What? We analyze the antigen antibody reaction. So in immunochemistry, we determine nothing but only and only antigen and antibody reaction. How they react with the antigen, or how the antibody react with antigen, and how the antigen react with antibody. So, okay. So immunochemistry is the branch of chemistry that involves the study of the component and function of the immune system, such as the nature and interaction of antigen and antibody. What? Antigen antibody. Immunochemistry involves the use of simple, rapid, robust, yet sensitive automated method for routine analysis in clinical laboratories. Okay, clear? Immunochemistry involves the use of simple, rapid, robust, yet sensitive automated method for routine analysis in a clinical laboratory. This is the picture how the immunochemistry will be takes place. This is a cell, this is the protein, This is antibody. How antibody react with this antigen? After the secondary antibody, that is labeled antibody, when they react with primary antibody, they produce a color. According to that color, we determine the antigen antibody reaction. In this, primary antibody are fluorescent tag. When they react with antigen, they produce a signal. What? They produce signal. So basically, this is a technique of the antigen antibody reaction. Now next is immunohistochemistry, that are already in a immunochemistry. This is covered in immunochemistry. This involves the use of monoclonal and polyclonal antibody. First of all, what is monoclonal and polyclonal antibody? Monoclonal antibody means when more than one type of antigen are present or and we want to isolate one type of antibody from that. If only one type of antibody we isolate and we clone that, that is known as monoclonal antibody. What? Monoclonal antibody. And for polyclonal, name clear. Poly means more than one type of antibody, means they are polyclonal antibody. So in immunohistochemistry, there is use of monoclonal antibody and polyclonal antibody. This involves the use of monoclonal and polyclonal antibody on histologically processed tissue section in order to spot specific antigens in a healthy or diseased tissue. Immunohistochemistry makes use of labeled enzymes such as peroxidase and alkaline phosphatase, and even collider gold at light microscopy and electron microscopy level in the diagnosis of cancer and surgical pathology. Already in previous slide, you see that how the second in indirect method, how the secondary antibody are labeled, and in fluorescence indirect method or fluorescence method, how the primary antibody are labeled with fluorescent molecule. So in immunohistochemistry also labeled antibody are used to determine the reaction between antigen and antibody. This is the method of immunohistochemistry. This is the substrate, right? This is the primary antibody. This is primary antibody. This is secondary antibody. This is peroxidase or alkaline phosphatase. This is color. See, this is protein, right? This is a protein. Okay. When this protein react with primary antibody, the secondary antibody, that is that labeled with a molecule which produce color, they react with primary antibody. When they react with primary antibody and we given that enzyme like peroxidase or alkaline phosphatase, they react with this substrate and produce a product, right? Produce a product, and color intensity will be generated. According to that color intensity, we determine the intensity or presence of antigen. What amount of antigen are present in our body according to that intensity. Now next, immunocytochemistry. This branch involves the microscopic study of cell sample. What? This branch involves the microscopic study of cell samples in a suspension or cell grown in a monolayer. Here the staining process is the same as immunohistochemistry, where in the enzyme catalyze the deposition of a dye at the antigenic site within the cell sample. In this, dye are catalyzed in the antigenic site, not in antibody site. In ICC, the expression of protein in a cell or group of cell also aids in understanding its intracellular localization. What? Understanding its intracellular localization. ICC assay are of two types: the direct ICC and indirect ICC. In indirect ICC, what happens? Secondary antibody are used, and indirect primary antibody are used, and that primary antibody are already labeled with a fluorescent dye. Clear? That are labeled with a fluorescent dye. See, this is the primary and this is labeled with a fluorescent dye. And here indirect secondary antibody are used, and that are labeled with a fluorescent dye. Now, what are the application of immunochemistry? Apart from the use of immunohistochemistry in identifying a prognostic marker in a cancer, it is used to predict response to therapy. Immunochemistry are used to know the response in the therapy, to identify infection as well as in muscular and neurodegenerative disorder. Identify infection as well as in muscular and neurodegenerative disorder. On the other hand, ICC, which involve the collection of cytological direct smear, is effective in identifying cancer such as metastatic marker, cell carcinoma, and in lung adenocarcinoma. Clear? ICC are used in metastatic marker, cell carcinoma, and in lung adenocarcinoma. These are the application of the immunochemistry. Detection of the virus in different tissue, indicating a role of this virus in the pathogenesis of certain disease in this tissue. Example: detection of EBV in patient with lymphoma and nasopharyngeal carcinoma. These are detected by using a immunohistochemistry. Detection of SCMV and rubella virus in the placental tissue of aborted or stillbirth babies. These are the application of the immunochemistry. Detection of SCMV in renal transplanted patient who had acute HCMV. This is the application of These are the application of immunohistochemistry. Now next is microscopy. What is a microscopy? Microscopy is the technical field of using microscope to view object and area of object that cannot be seen with the naked eye. Remember this. The substance or microbes which are not seen by a naked eye, that are seen with the help of microscope. From name it is clear, micro means small. What? Small. Right? We see these small things into a large form. And these are converted with the help of microscope, because microscope contain a lens that increase or magnify the size of particles or object. There are three well-known branches of microscopy. That is optical microscope, electron microscope, and scanning probe microscopy, along with the emerging field of X-ray microscopy. These are a field of the microscope, that is optical microscopy, electron microscopy, and scanning microscopy. Actually, microscope are used in every branch, like in microbiology to see the microorganism, in biochemistry to see the sample, to see the microorganism that are present in a sample, to see the particles in clinical to see the particle or particles, molecules, substance that are present in a sample that are collected with the technician, like in urine. In urine we want to see the crystals, microbes, that how we see these things with the help of microscope. So they are huge application of the microscope. What huge application of the microscope? This is the diagram of microscope. This is the general diagram of the microscope. This is the ocular lens, from where we see the object. This is the revolving nose piece. This is the objective lens. This is a stage. This is iris diaphragm. This is condenser. This is light source, and this is a base. Actually, here we take a sample or place a sample, and from this is a condenser. From condenser, this is the objective lens. This is the objective lens. We collect this sample by this lens. After that, by magnifying by the lens, we see this by a ocular lens or eyepiece lens. Clear? This is a coarse adjustment. How we adjust the core? Core means location of the sample. We want to move the sample by adjusting this core. And this is the revolving nose piece. Means if you want to, there are 4 types of objective lens are present in this one microscope: 10x, 20x, 40x, like this. So according to your object or sample, we adjust this lens, objective lens, by revolving the nose piece. Clear diagram? Now first, what is the dark field microscope? From name it is clear. Dark field means the object appear bright in a dark background. See here, the background is dark, right? What? Background is dark here. What? Right? In dark background, the sample will be bright here, or object will be bright. So that's why the name is dark field microscopy. From name it's clear. So remember this. In dark field microscopy, the background will be dark and the object will be bright. Clear? What is the principle of dark field microscopy? In dark field microscopy, the object appear bright against a dark field. This is made possible by use of a special dark field condenser. What we use in this? In this we use a special type of dark field condenser. What is the application? It is used to identify the living unstained cells and thin bacteria. This is the techniques, this is the dark field microscopy by which dark field microscopy by which we identify the living unstained cell and thin bacteria like spirochetes, which cannot be visualized by light microscopy. Clear? Now next is bright field or light microscopy. From name it is clear. In dark field, what happens? The background will be dark and object will be light. Same here. In bright field microscopy, the background will be light. The background will be light and background will be bright and object will be dark. Opposite of the dark field microscopy. What is the principle of the bright field microscopy? The bright field or light microscopy form a dark image. What? Form a dark image. This is the dark image against a brighter background. This is a brighter background. Microbiology and bacteriology have also relied on the bright field techniques. This bright field microscopy are also used in microbiology and bacteriology. Different is how this object are seen as a dark color, because something we stain the object, that's why this object are look like dark in color. So in this techniques staining method are used. Different types of stain and staining techniques are used depending upon the type of specimen. Depending upon the type of specimen and cell structure being examined. Which type of specimen are used and what type of cell specimen are examined under the microscope. According to that only, bright film staining method are used for the sample. What type of cell are examined in bright field microscopy? Fuchsin stain is used to stain smooth muscle cells. What? Fuchsin stain. Gram stain is used on a bacteria, right? Already know gram stain are used for the bacteria. Gram stain is used on bacteria and give rise to a name gram negative or gram positive bacteria based on the reaction of the bacteria to the stain. Clear this? Now, what are the difference between bright field microscopy and dark field microscopy? From name, first difference is from the name. Bright field means background is bright and dark field background is dark. Light from a plane wave source is focused through an object by a condenser in bright field, and in dark field an opaque disc is put between the source and the condenser, blocking out the middle of the beam. Some light is blocked by opaque part of the object or reflect away at the boundaries between component or material of different refractive indices in bright field microscopy. In dark field, the main role of the condenser, and in bright field main role of the staining technique. So in dark field, condenser focus the beam onto the sample. In bright field, the remainder passes through the objective lens to the observer. In bright field, this procedure, a bright background with object details appearing darker in the image than their surrounding. The brightness of the image is determined the source brightness and block size. This results in poorer contrast compared to dark field, as the dark area are generally grey rather than black. In dark field microscopy, only light scattered by the object enter the objective. This procedure this produces a dark background with sample detailed appearing brighter than surrounding. The brightness of the brightest part of the image is determined by the amount of light scattered by the object. So in simple language, what is the if someone asks you what is the difference between bright field and dark field, then you clearly say that first difference sorry first difference is in bright field microscopy, bright background is used. Background is bright. Background is bright. In this, background is dark. Second difference, staining techniques is used. In this, unstained sample is used. Third, dead sample is used. In this, live sample is used. Fourth difference, in this object we see is darker, and in this the object we see is brighter in color, right? So these are the simple difference. In this, mainly function of condenser depends. Functioning of condenser produce dark background. In this, staining techniques used a dark object. Okay, these five are the main difference. It's main and simple difference between bright field microscopy and dark field microscopy. Okay, clear? Now next is phase contrast microscope. In phase contrast microscope, actually in dark field also, right? In dark field also living cells are used, and in phase contrast also living cells are used. It is used to visualize the living cells by creating differences in contrast between the cells and water. See here, living cells. This is the living cells that are used to visualize difference between the cells and water. It converts slight differences in refractive index and cell density into easily detectable variation in light intensity. It is useful for studying microbial motility. These techniques are used for the study of the motility. Study the motility. And second, what are the application? Determine the shape of the living cells. So mainly phase contrast microscope are used for the living cells to know the shape and motility. To know the, if some question are asked, which techniques are used for the living cells to determine the motility, then the answer is phase contrast. Now next is electron microscope. In electron microscope, from name it is clear, electron beam are used. What? Electron beam are used in these techniques. It was invented by Ernst Ruska in 1931. It differs from light microscope by various ways. There are two types of electron microscopy: transmission electron microscopy and second is scanning electron microscopy. Transmission electron microscopy are used to examine the internal structure, and scanning electron microscopy are used to examine the surface. This is the same, that is scanning microscopy. This is used to scanning the surface. And this is the TAMP, that is transmission electron microscopy. This is in this internal structure we also see. So TAM is used for internal structure and SAM is used for surface determination. Scanning electron microscopy allows scientists to view a universe too small to be seen with a light microscope. SAM do not use light waves, they use electron, negatively charged electrical particles, to magnify object up to 2 million times. What? Object up to 2 million times. SAM creates a 3D view of a specimen but cannot view living specimen. SAM creates 3D view of a specimen but cannot view living specimen. Right? In this technique, living specimen are not used. Transmission electron microscopy also uses electron, but instead of scanning the surface, electrons are passed through the very thin specimen, and TAM are used to see the inside or examine the internal structure of the cell, not only the surface. Clear? TAM is used for examining the internal structure of the surface, whereas SAM is used to examine the surface only. Now, another type of microscopy is also, that is two-photon microscope. Two-photon microscopy is also a laser scanning microscopy, but instead of ultraviolet, blue or green laser light, a pulsed infrared laser is used for excitation. Which type of laser light are used in two-photon microscopy? Blue or green or pulsed infrared laser is used for excitation. Only in the teeny focus of the laser is the intensity high enough to generate fluorescence by two photon excitation, which means that no out of focus fluorescence is generated and no pinhole is necessary to clean up the image. Clear? In two photon microscopy, laser beam are used. What? Laser beam are used in this. And only in the teeny focus of the laser is the intensity high enough to generate fluorescence by two photon excitation, which means that no out of focus fluorescence is generated and no pinhole is necessary to clean up the image. Okay, in two photon microscopy, we see the object with the help of laser light. Okay, so today we stop here. In this video we cover all the analytical part. Okay, hope
Hello students and welcome to the study portal. The topic of today's online video is serological tests and techniques. So in today's online class, we are going to study about introduction to the serology, that what is serology, and the different tests and techniques that are used in the laboratories and the hospitals for the diagnosis of different patients. So what are the learning objects of today's class? Let's have a look at that. So at the end of the lesson, you should be able to understand or list different types of serological tests. You should be able to state the principle of serological tests, describe the application of different serological techniques, identify the advantages and drawbacks of different serological techniques, and identify the factors which affect the antigen antibody reaction. So let's start with the introduction to the serology. So what is serology? So serology is the scientific study of serum, the body fluids. If you will look at the word, it is made up of two root words. That is, first one is the sero and the second one is the logy. The logy words means study, whereas sero means serum. So you can state that the study of serum is defined as the branch of serology. So, if we talk about practical terms, it is usually the diagnostic identification of antibodies in the serum. It is also the tests that are performed in the serology are usually called as the serological test. Alright, so they are generally also called as the antibody test. So many of the antibodies, as you all are aware, that they are present in our your body in response to any infection or any kind of like disease or bacteria or any kind of pathogenic agent. If it enters into our body, then what our body do, what our bodies generate the antibodies to fight with that kind of infection, to stop that foreign particle or protein to develop or grow in our body, right? So what are serological tests? The serological tests are the serology test or antibody test. These are any of several laboratory procedures that are carried out on a sample of blood serum, we can say. What is serum? Serum is the clear liquid that separates from the blood when it is allowed to clot, right? When these laboratory procedures are carried out on a sample of blood serum for the purpose of detecting antibodies or antibody-like substances that appear specifically in association with certain diseases, these tests are called as the serological test, right? This is clear to you. So there are different types of serological tests that are performed in the laboratories. These are flocculation tests, the hemagglutinin inhibition test, ELISA, RIA, you all know about that, right? So the serological test is particularly helpful in the diagnosis of certain bacterial, parasitic, and viral infection, including many of the infections such as yellow fever and infectious mononucleosis. It is also useful in the detection of autoantibodies that those are harmful antibodies that attack our body or the components of our body that are involved in the autoimmune diseases, right, such as rheumatoid arthritis. All right, so serological testing, we can say, has proved very valuable in the detection of diseases such as syphilis, HIV or AIDS, and epidemic and pandemic infections also. So these are the applications of the serological test, that they have an diagnostic, they are performed for the diagnostic process, such as the diagnosis of any kind of infection or any kind of autoimmune diseases as well. All right, so they are also detected in patient with certain immune deficiency who are associated with the lack of antibodies. Now, let's have a look at the introduction or the general principle of the serological test, on what principle these serological tests are based upon. So, the serological reactions or serological tests are based on the antigen or antibody interactions that undergo in the human body. We can say that the antigen reacts with antibody, and then an antigen antibody complex is formed, and these antigen antibody complex can be seen in such as precipitation, agglutination, flocculation, etc. So the detection and measurement of these reactions, these antigen antibody interaction or reaction, form the basis of the serology, which is a subdiscipline of the immunology. Therefore, we can say that serology is the science of measuring antibody or antigen in the body fluids. Talking about the immunological techniques or the kind of tests that are performed in the under the branch of serology, so these are also called as the immunological techniques, because the immune systems, antigen and antibodies are involved in these type of reactions. So basically there are three types of tests that are usually performed in the immunological techniques. First one is the primary binding test, second one is the secondary binding test, and the third one is the tertiary binding test. In this video, we are going to focus on the primary binding test only, and the rest of test, the secondary binding test and the tertiary binding test, we will deal with it. We will focus it in the next video, in the next part of this video also. Alright, so in first part, we are going to talk about the primary binding tests that are performed in the laboratory. So the example of primary binding test is ELISA test, that is enzyme linked immunosorbent assay test, that is widely performed in the laboratory. Second one is the RIA, which is radio immunoassay. Third one is the Western blotting, that is also performed for HIV, AIDS diagnosis. The next one is the Northern blotting and the Southern blotting. And the fluorescence tests are also performed under primary bindings test. So we are going to study about the all these listed tests in today's session, right, students? So what are primary binding tests? So primary binding tests are tests that directly measure the binding of antigen and antibody. We can say that it directly measures or visualize the immune complex. So these are the most sensitive techniques in terms of the amount of detectable antigen or antibody. What are the example of primary binding test? I have given you the example. Examples are ELISA, RIA, Western blotting, Northern blotting, and the Southern blotting. So primary binding tests are performed by allowing antigen and antibody to combine and then measuring or visualizing the amount of immune complex that is formed. It is usual to use certain kind of radioisotopes, fluorescent dyes or enzymes as labels to identify one of the reactants. Alright, so these are widely used in the serological diagnosis of bacteria, virus, fungus, and the parasites. So these are usually sensitive tests, and they also give reproducible results. So talking about immunofluorescence test. So immunofluorescent test, what is it? Immunofluorescent tests are widely used tests in the serological diagnosis of bacterial, viral, fungal, and parasitical disease. They are usually sensitive kind of tests. All right, so what is the principle of this immunofluorescence test? Let me tell you. It is based on the fluorescent dyes, use of fluorescent dyes, or we can say the fluorochromes, which are illuminated by the UV lights that are used to show the specific combination of an antigen with its antibody. So the antigen antibody complexes are seen fluorescing against the dark background. The immunofluorescent tests are also referred to as the fluorescent antibody test or FAT, right? So we can say that the immunofluorescence tests are of two types. First one is the direct fluorescent antibody test, and second one is the indirect fluorescent antibody test. Now talking about direct fluorescent antibody test. So direct fluorescent antibody test is used to detect and identify an unknown antigen in the specimen. It could be a virus, bacteria, or the parasite antigen. Why it is called direct now? Why it is called as direct fluorescent antibody test? It is called because the fluorescent dye is attached or labeled directly to the antibody. The fluorochrome dye, which is usually used, is the fluorescein isothiocyanate, which gives a yellowish green fluorescence to the component. So the fluorescent substance is the one that when absorbing light of one wavelength emits light of another longer wavelength. So this is how the direct fluorescent antibody tests are performed. So what is the procedure of direct FAT? Let's talk about it. So a tissue or a smear containing the organism is fixed to a glass, right? And then it is incubated with the fluorescent antibody. Then it is washed to remove the unbound antibody. Then it is examined by dark field illumination under a microscope with a UV light source. So the antigenic particles that have bound the labeled antibodies are seen to fluoresce brightly, right? If there is presence of fluorescence, it means that the test is positive for that particular antigen. If there is no fluorescence, then the test is negative or absent of particular antigen. So why direct FAT test is used? It is used to identify bacteria when the numbers are very low, and also to detect viruses which growing in tissue cultures or tissue infected from infected animals such as rabies virus, etc. Now talking about the indirect fluorescent antibody test. So in the indirect fluorescent antibody test, unlabeled antibody combines with the antigen, and the antigen antibody complex is detected by attaching a fluorescent labeled anti-species globulin to the antibody. So what happens, the antibody therefore is labeled indirectly in this process, because the antibody is not directly labeled with the antigen. Otherwise it is attached to a fluorescent labeled anti-species globulin, right? So fluorescent labeled anti-human globulin is used if the antibody is of human origin, we can say. Now it is used in two ways: to detect and identify unknown antigen in the specimen, and to detect antibodies in a patient serum using a known antigen or microorganism, right? So in indirect fatigue, to detect antigen, what happens? A slight preparation of specimen is made and unlabeled specific antibodies are added to after incubating the so that the antigen and antibody can combine. The preparation is washed, leaving only antibody that has combined with the antigen on the slide. Thereafter, a fluorescent labeled anti-species globulin is added and allowed to combine with the antibody. Then the excess is washed. Then the preparation is examined under fluorescent microscope. Right. If the fluorescent is seen, it means that the particular antigen is present. Otherwise, the antigen is absent. Now talking about the ELISA, which is enzyme linked immunosorbent assay. So ELISA techniques are becoming increasingly used in the diagnosis of microbial infections. They are specific, sensitive, and require only a small amount of specimen. So these are the like we can say that the reagents used in the ELISA are stable and have a long shelf life, which makes it for easy distribution to district laboratory. The results also of quantitative qualitative ELISA techniques can be read visually. Large number of specimens can be tested at one time, and the ELISA can be easily automated for the use in epidemiological services surveys. So moreover, we can say that ELISA are suitable for automation, objective reading of results, and the ability to link ELISA plate readers to the laboratory computer system, which also reduces the potential for the transcription errors, right? Now talking about the types of ELISA. So ELISA can be performed as double antibody technique, which is performed to detect the antigen, and the indirect technique, which is used to detect and assay the antibody, right? So let's talk about the principle of ELISA. So as the name suggests, the ELISA, which means the enzyme linked immunosorbent assay, uses an enzyme system to show the specific combination of an antigen with its antibody. The enzyme system consists of an enzyme which is labeled or linked to a specific antigen or antibody. It also consists of a substrate which is added after the antigen antibody reaction. We can say that the substrate is the food of the enzyme. This substrate is acted on by the enzyme attached to the antigen antibody complex, which gives it a change in the color. Then what happened? The intensity of the color gives an indication of the amount of bound antigen or the antibody. Here also you can see in this picture that the antibody coated well is there. Then we will add antigen to be measured like specimen is added to this coated wall. Well, it will bind to the particular antibody. Then it is again washed, and then we have to add enzyme conjugated secondary antibody. It will add, and we will wash. Then if the substrate is particular to the enzyme, the color of the this solution will change, and the intensity of the this color is measured to quantitative results, right? This is clear. So talking about the indirect ELISA, right? So what is indirect ELISA? So in indirect ELISA technique, known antigen is attached to the inside surface of the well, and patient serum is added to it. After incubation and washing, the enzyme labeled anti-human globulin is reacted with the antibody which is attached to the antigen. The uncombined labeled enzyme is washed from the well, and a substrate is then added. Then the presence and concentration of the antibodies which has reacted with the antigen is shown by a change in color of the substrate. The more intense the color is, the higher the concentration of antibody in the serum, right? So this is how it is performed. So the indirect ELISA is, we can say, is used in the diagnosis of several parasitic infection and is being increasingly used in the diagnosis of bacterial, fungal and HIV infection also, right? This is clear. Now talking about the RIA, which is radio immunoassay. So radio immunoassay is a competitive immunological test which is performed for measuring very low concentration of the antigens or antibodies by using the radioactively labeled antigens as the competitors, right? So the radioactive isotopes such as tritium or any kind of this can be used for the labeling. So RIA is a highly sensitive method to detect low concentration of the unknown antigen and is also used to assay compounds such as hormones, drugs, enzymes, microbial antigens, example hepatitis B antigens, CEA and alpha fetoprotein antigen. It is also used for the detection of antibodies. RIA was first developed by the endocrinologist S.A. Burson and Rosalind Yellow in 1960 to determine levels of insulin anti-insulin complex in the diabetics. Right. Now talking about the components that are used in the RIA technique. So generally three components are used in the RIA technique. These are patient antigen, which is the specific compound that we wish to determine, that we have to determine. Second one is the labeled antigen, which is the same compound, the patient antigen, which is attached to a radioactive label, and the antibody which are specific for the sample and labeled antigen, right? So the RIA can be performed in two ways. First one is the liquid phase assay and second one is the solid phase assay, right? So what happens in liquid phase? In this assay, in liquid phase assay, the sample labeled antigen and the specific antibodies are added to a mixture in a solution form. After completion of incubation with the ligand of interest, a bound-free separation step is performed using different techniques. Whereas if we talk about the solid phase, the specific antibodies is added either in the suspension or the antibody is covalently bound to the inside wall of the reaction tube. Then the separation of the bound free fraction is realized by centrifugation or magnetic separation, which is followed by decanting the supernatant, or we can say by simply pouring of the reaction mixture if the coated tube is used, right? Now talking about the IRMA, which is immunoradiometric assay, which is also called as the sandwich immunoassay. So why IRMA is there? For IRMA is there for the physical separation methods, centrifugation or decanting procedures, we can say, that may distort the equilibrium between bound free fractions. Sledge loss of labeled antigens can be there. Frequent non-specific binding can be there, and the lack of sensitivity for very low concentration levels of a particular analyte can be there. So the basic principle of IRMA is based on the specific antibody of the sample antigen is bound to the inside wall of tube and the tube is coated with antigen when a specific antibody is to be determined. Thereafter, by following the process, labeled antibody or antigen are used in which the same specific antibody is labeled with the radioisotopes, right? And the result is perform. Now talking about the western blot. So western blot is generally used as a confirmatory test as a confirmatory test, and it is very specific for the HIV test. In the case of HIV, it is like confirmatory test and it is very specific. It separates the blood proteins and detects the specific proteins that indicate an HIV infection, right? So western blot test is typically used, we can say, to confirm our positive HIV diagnosis. During this test, what happens? A sample of blood is taken and it is used to detect HIV antibodies. The Western blot test separates the blood proteins and detects the specific proteins that induce any kind of infection. It is used to confirm a positive ELISA, and the combined tests are 99.9% accurate, right? So we can say that it is an antibody test that is done on the blood sample, right? So these are also known as the protein immunoblots. These tests are used to detect specific proteins in a sample. Now talking about the southern blotting technique. So the southern blotting technique was discovered by the Southern scientist who first described it in the 1975. It is a method which is used in molecular biology for the detection of specific DNA sequences in the DNA sample. Southern blotting combines transfer of electrophoresis separated DNA fragments to a filter membrane, and then subsequent fragment detection by prone hybridization is performed. Right? So other blotting methods are also used along with it, but using RNA or protein have later been named in reference to the Southern's name. So this is how the southern blotting is performed. It is used in clinical lab to detect gene mutation which are known to cause any kind of diseases. It is also used to detect carriers of the fragile X syndrome, and it is generally used to detect specific DNA sequences. So students, that's about the primary binding tests that are performed in the serological test. Hope you are able to understand the concepts of the serological test, that what are serological tests, what is a serology, and the different types of primary binding tests that are performed in the laboratory for the diagnosis of any kind of. Hello students and welcome to the study portal. Hope you all are having a very good day and may your day be as bright as the sun. So the topic of your the today's online session, as it is flashed on your screen, is serological test and techniques part 2. So in today's session we are going to discuss about the part 2 of the serological test and techniques. It is in continuation with the serological test and techniques part 1, which was about the serological test introduction, that what are serological tests and what are the different kind of tests that are performed in the serology. So let's have a quick sneak peek at the review of the first part. So in the first part, we have studied about the serological test, its introduction, that what are serological tests and how what is serology, what is the branch of serology. We also discussed about the different types of serological tests that are performed in the laboratory. We mainly focused on the primary binding tests that are performed in the serological test in the laboratory. The examples of primary binding tests were ELISA, RIA, northern blotting, western blotting, etc. In today's session, we are gonna discuss about secondary and tertiary serological tests. So at the end of the session, you must be able to explain secondary and tertiary serological tests. You will be able to differentiate between different types of secondary and tertiary serological tests. You will be able to state the principle of these tests and the applications of different serological techniques. You'll be able to identify the advantages and drawbacks of different serological techniques and the factors which affect the antigen and antibody reaction. So what are immunological techniques or the serological tests? So these are done or performed in the laboratories to detect and measure the antigen antibody combination. So, what is serology? Serology is the branch of science which deals with the study of the serum. The in immunological techniques or immunological tests, what we focus upon is the antigen antibody interaction. So, in these kind of tests, the antigen and antibody interact with each other and form a antigen antibody complex, which is then visualized or under the microscope or with naked eye, or will be used for the quantitative detection of the particular antigen or antibody that is present in the particular sample, right? So there are three types of immunological techniques, or are the three classes of immunological techniques that are widely used in the laboratory. These are primary binding test, secondary binding test, and the tertiary binding test. So the primary binding test we have covered in the first part of the video. In today's session, in today's video, we are going to discuss about the secondary binding test and the tertiary binding test. So what are secondary binding tests? So the secondary binding tests, we can say, are the tests that detects and measure the consequences, or we can say the secondary effect of the antigen antibody interaction. What all these consequences can include? It can include the precipitation of soluble antigens, the clumping of particulate antigens, we can say the neutralization of bacteria, viruses, or toxins, and the activation of the complement system. So we can say that the secondary binding tests are usually less sensitive than the primary binding test, but these are easier to perform. That's why they are in vogue in the laboratory. So first such kind of test, the secondary binding test, is agglutination test. So agglutination tests are frequently used in the laboratory, because if we will compare these with the other serological tests, these are simpler to perform, they require no specific equipment, and these are usually less expensive, right? So what is the principle of these agglutination tests? So agglutination is the visible clumping together of bacteria, cells, or particles by an antigen combining with its specific antibody. What happens? The clumps will form, and the resulting clumps are referred to as the agglutinates, right? So it when it will agglutinate, it will show cells with surface antigen being agglutinated by the specific antibody. Why this occurs? This occurs when using specific antisera to identify bacterial colonies. It is also possible for free antigen to agglutinate particles that have been coated with the specific antibodies. This occurs when testing CSF for extra soluble cellular soluble antigen. In agglutination tests, these are used to detect antibody in a patient's serum. A known antigen suspension is used. The antigen particles are agglutinated if the serum contains the corresponding antibodies. In general, to detect antibody in patient serum, a known antigen suspension is added, or to detect antigen in the serum, a specific antibody is added. A good example of secondary binding test, or in this classification agglutination test, is the blood grouping test. In blood grouping test, we don't know the particular antigen. So what we will do, we will particular antibody. So what we will do, we will add the monoclonal antibodies that are there. We will add the monoclonal antibodies with the person's blood, and then it will combine the antigen and antibody will react, and it will give us the clumping, and the clumping is observed under the microscope and with the naked eye as well, and then the results of the blood group are given to the patient, right? So agglutination test can be performed on slides or tiles, in tubes, and in micro titration plates as well, right? So what are slides or tiles agglutination test? So the slides or agglutination test, we can say that it can be rapid. These are rapid, these are easily performable techniques which give a reaction in minutes or even seconds. They are we can say they are however not usually as sensitive as tube or micro titration technique, but their specificity depends on the reagent that we are using it for it. The type of agglutination that can be observed on these kind of slide or tiles agglutination test is active or passive. Right. Now talking about the tube agglutination test. Right. So in tube test, agglutination occurs in a larger volume of fluid, and therefore we can say in an environment that can be more fully controlled. It provides that kind of environment. So tube tests are usually more sensitive than the slide test, right? So in this tube agglutination test, what happens? A serum is diluted serially, and then antibody level is measured by adding standard antigenic suspension. In the tube test, the temperature and time of agglutination must be correct, and a control tube that contains only the antigen suspension must be included to check for agglutination of the reagent, right? So the tube tests are used mainly to detect and measure serum antibodies in the investigation of enteric fever or brucellosis or any kind of such diseases, right? Now talking about the micro titration agglutination test. So micro titration agglutination tests are performed in the micro titration plates. They have now replaced several tube agglutination tests since they are more sensitive, more economical. These are easier to perform and usually give quicker results, right? So the type of micro titration agglutination test is indirect hemagglutination test, which is also called as the IHA, right? And hemagglutination inhibition antibody test, which is also called as the HIA, and the reverse passive hemagglutination test also, right? Discussing about the hemagglutination test, indirect hemagglutination test, which is also termed as IHA. Indirect hemagglutination test is a passive, we can say passive agglutination test in which known antigen is coated on treated red cells, right? So, application of the IHA includes the treponema pallidum hemagglutination to detect the treponemal antibody, and the anti-streptolysin O titration technique ASO test, which is used in the diagnosis of S. pyrogens infections. Right. Now talking about the hemagglutination inhibition antibody test. So what is hemagglutination inhibition antibody test? It is used to detect antibodies against arboviruses, influenza viruses, measles viruses, and the rubella viruses. What these viruses are able to agglutinate red cells because they possess hemagglutinins on their outer surface. So what happens in this hemagglutination inhibition antibody test? The patient's serum is reacted with the suspension of known viral antigen. If the corresponding antibody is present, it will coat the hemagglutinins on the viral particles, and it will prevent hemagglutination when red cells are added to it. Controls are usually used with it to show the agglutinating activity of the antigen and the absence of hemagglutination by the serum. Right. Now talking about the second types of secondary binding tests, that are the precipitation tests. The precipitant techniques are used to detect and identify antigens in the specimen, extracts, and cultures, and also used to detect and quantify antibodies in the serum. If we will compare it with agglutination tests, the precipitant techniques require more experience in their performance and interpretation. Right. The precipitation tests are based on the principle that the antigen and antibody are in a soluble form and combine to form a visible precipitate. The presence of electrolyte is usually required for these kind of tests, right? Positive and negative controls are also very essential for performing these tests. So various systems are available in which the precipitation tests are performed. They can be performed in semi-solid media, which is agar or agarose, or non-gel support media as well, such as cellulose acetate. Right? So these are used to detect and identify antigen in specimen, extracts, and culture. Right? These tests have a low sensitivity. So there are different types of precipitation tests that are performed in the laboratory. First one is the tube precipitation test. Second one is the gel diffusion test, and third one is the counter immunoelectrophoresis test. Let's discuss about all of these in details. The first one is the tube precipitation test. So what happens in a tube precipitation test? In tube precipitation test, a clear solution containing the test antigen is carefully layered onto a clear antiserum in a precipitant tube, or we can say a capillary tube. Then following a period of incubation, if the corresponding antigen is present and the proportion of antigen to antibody is optimal, a line of visible precipitating antibody and antigen will form between the two layers of the fluid. A fairly large volume of antiserum is required, and this test is not very sensitive. So examples of the tube precipitation test are the testing of CSF for extracellular antigen, and the original lens field method of grouping beta hemolytic streptococci. Now discussing about the gel diffusion test. So when both antigen and antibody diffuse through the agar, this is referred to as double diffusion. When only the antigen or antibody diffuses with the corresponding antigen or antibody being coated in the agar, this is called single diffusion. So several hours of diffusion are needed in the gel diffusion before the precipitation may occur. So this is also a long time taking procedure in which most of the time is consumed, right? So it is used to detect and measure immunoglobulins in the serum and other specimen. For example, the detection of IgM in CSF when investigating the meningoencephalitis, etc. All right. Now talking about the counter immunoelectrophoresis test. The counter immunoelectrophoresis test is also called as the counter current electrophoresis or the immunoelectroosmophoresis and the electroimmunodiffusion. Right. So what happens in this test? In this kind of test, the electrophoresis is used to increase the speed with which the antigen and antibody travel in the agar gel. What happens? A line of this precipitation forms where the two meet in the optimal proportion. The pH, purity, and the ionic strength of agar are may influence the amount movement of antigen and antibody. That thus they are maintained in a controlled environment. If we talk about the principle of this counter immunoelectrophoresis test, then we can say that it is based on the principle of immunoelectrophoresis. What happens? Specific antibody is placed in a well at the positive electrode end of the plate, and the unknown antigen in a well at the negative electrode end. So, electric current is applied, and the antibody and antigen will move towards each other. So the positive sample shows a line of precipitation within 30 to 60 minutes. So counter immunoelectrophoresis gives more rapid results. It is usually more sensitive, but it has a disadvantage of requiring an electrophoresis unit in and the preparation of an agar gel. So CIE is generally used to detect extracellular antigens in the CSF and the detection of hepatitis B surface antigen in the serum. Now talking about the complement fixation test. So what are complement fixation tests? These are the third type of secondary binding tests. They are best performed in the reference laboratories where the facilities exist for the careful standardization and control of the reagent, because it requires a lot of control and the standardization. So we can say that this complement fixation test is a technique which has been used for many years to quantify antibodies that does not agglutinate or precipitate when these are reacted with its antigen, but it can be demonstrated by its use or fixation of the complement. So it consists of two types of component. First component is the indicator system, which consists of combination of sheep red cells, um, complement fixing antibody produced against the sheep red cells in the in another animal, and exogenous source of complement. Usually, it can be a guinea pig serum. So when these three components are combined in optimum concentration, the anti-sheep cell antibody complexes, which may cause cell lysis. The second component which is used consists of a known antigen and inactivated patient serum. So in the CFT, the patient's inactivated serum is serially diluted and reactively known antigen in the presence of complement. If the corresponding antibody is contained in the serum, it will combine with the antigen and use up the complement, which will leave no complement to hemolyze the antibody coated red cells which are added. So the highest dilution of serum that prevents hemolysis the antibody titer. So if the patient serum does not contain the corresponding antibody, the complement will not be used and will be available to fix and hemolyze the antibody coated red cells. Right? This is how the test is performed in the laboratory. Now talking about the tertiary binding test. So tertiary binding test measures the consequences of immune response in the in the human body. So these tests are much more complex, we can say, than the primary and secondary test, but their result reflects the practical significance of the immune response. For example, we can measure the protective effects of antibody also in these kind of tests, but these are not usually performed in the laboratory. Now let's discuss the factors which affect the antigen antibody reaction. So there are many factors that affect the interaction between the antigen and antibodies. They may include the specificity, cross reactivity, temperature, pH, ionic strength, and the concentration and intermolecular specificity. So the specificity, what is it? It is the ability of a particular antibody to contribute to combine with one antigen instead of one another, is referred to as its specificity. So it depends on the antigen antibody antigen binding fragment of immunoglobulin molecule and the antigen antibody reaction that can show a high level of specificity. Right. So these are certain factors that impact or that affect the interaction between the antigen and antibody. So that's all for today, students. If you have any query or doubt regarding this session, you may write in the feedback section on your LMS. That's all for today. Hope you understood the topic very well. Please go through both the parts of the video. The part 1 includes the introduction to the serological test and the different its classification and the primary binding test, which includes very common tests such as ELISA, RIA, Western blotting, Northern blotting, and the Southern blotting. But in this part, we have covered about the secondary binding test and the tertiary binding test that are performed, right? So there are different types of secondary binding tests also, such as the agglutination test, flocculation test, precipitation test, etc. Hope I'm able to clarify all your doubts regarding this topic. Thank you for watching, students. That's all.